Okay, let's see what we got here in terms of joining sugars together. Now we join sugars together by classic condensation type reactions. So I'm going to draw a sugar here. Once again, these detailed structures are not particularly necessary for you to remember unless you take it by. We do that in biochemistry, but we won't do that here. Okay, I'm not going to draw the whole thing, but let's put another sugar here. Another 6 carbon sugars. And I'm not going to draw the rest of that either. But what we see here, we have lots of hydroxyl groups and lots of hydrogens coming off the carbons and sugars. So those are great ingredients to make a condensation reaction. So let's do one here. What we will do, once again, is remove this OH group and this hydrogen, release it as water, and then form a covalent bond directly between the carbons. And that's how we join sugars together.
You said that's condensation?
Yes, or dehydration or whatever. Okay, and once again, that is an endergonic energy requiring process. Now, that's not the only way we can do it though, because notice this molecule, I haven't drawn all of it, has tons of OH groups and tons of hydrogens coming off of these carbons. So there are other ways we could join the same 2 sugars together. Now, I'm going to do the shorthand notation here, because I just don't want to draw the whole thing out. All right. So what we did here was make a covalent bond from here to here. However, there's no reason we couldn't make a covalent bond, say, from here to here. Instead, like this. So, or even from something like from here to here. There's many different ways of covalently bonding the same 2 sugars together. Yes?
On this left molecule,
Yeah.
what happens to the hydrogen that's bonded to the carbon? Because carbon has 5 bonds there.
Okay, sorry. It's not supposed to have a hydrogen. Okay, sorry about that. You are absolutely right. It's not supposed to have 5 bonds. Okay, sorry. All right, so, okay. Hydrogen never existed. Okay, thanks. Thanks. All right. Well, anyway, and likewise, we would have it. Okay. So we could do a covalent bond from here to here, or from here to here, and so on. So there's many different ways of making covalent bonds from one sugar to the next. As a matter of fact, this is a property that is unique to polysaccharides. We could have a couple sugars joined together like so. All right. But over here, we could make a second covalent bond to a totally different sugar. Like so. Oops, make sure I draw it right. And we can continue that process on. So what we just done is this sugar has not just one other sugar, but 2 other sugars bound to it using different covalent bonds. We've made what would be ordinarily a straight chain of sugars. We've developed a branch point over here. And you could have a polysaccharide, straight chain, and then a branch comes off, and then maybe another branch comes off, and you end up with a branch structure like a tree. That is something, and the reason we can do that is we can have one sugar make 2 different covalent bonds to 2 different sugars generating a branch. Now, that is a property that is unique to polysaccharides. None of the other biological molecules, big biological polymers, can do that. Proteins, amino acids, are only one way of joining amino acids to each other. There's only certain ways of there's only one way of joining nucleotides together to make nucleic acids. So all those molecules are straight. They're long, straight molecules. But here, polysaccharides can form branch chains. And they are the only large biological molecule that can do that. Now, why is that important? Recall we said that certain polysaccharides serve as molecular recognition markers. In other words, they're like flags or labels on other types of molecules. Well, you might need an awful lot of flags because there's an awful lot of different types of molecules and an awful lot of different types of cells around. And in order to do that, you need a lot of diversity. Now, the thing is, there aren't that many sugars, maybe about a dozen or so sugars that are commonly used in polysaccharides. So there's not too many different types of sugar subunits that are widely used. But if you can make chains that can branch in all kinds of different shapes and directions and things like that, that gives you a tremendous amount of structural diversity. And you can, and virtually infinite, and you can use that to make a whole bunch of different recognition markers. So that's actually an important property of polysaccharides. So we'll say structural diversity. And once again, the ability to form branch chains like we have here is unique to polysaccharides. No other large biological molecule can do that. All right. So we join two different sugars together. Now, by the way, we're going to see this very shortly. The different types of covalent bonds. You can have a polysaccharide. You can have different polysaccharides made of the same type of sugar. Just one single type of sugar and many copies of it. And those polysaccharides will have different properties and different biological roles. Even though they're all made of the same kind of sugar, they can have different properties because the key thing is just what types of covalent bonds are used to join them together. Now we're going to see that coming up fairly soon. Not quite yet, but coming up pretty soon. Okay, questions on that? Before we go on to polysaccharides, let's take a look at a few common, what we call disaccharides. Because a number of organisms will produce these disaccharides. They'll covalently bond two sugars together and then use them for storage or whatever the case may be. So disaccharides are two sugars covalently bonded together. Okay, the one that we're most familiar with, you see on your breakfast table every morning. That's sucrose. And what sucrose is a glucose molecule covalently bonded to a fructose. All right. Now, of course, that's widely used as a flavoring ingredient. You know, when you have all kinds of candies, cookies, the whole 9 yards. All kinds of, you'd be surprised what kind of things have sugar in it that you don't know about until you read the ingredient label. And it's very widely used. Now, it turns out that sucrose is produced only by a couple different kinds of plants and stored in large quantities. The most common one is a type of grass called the sugar cane. And the sugar cane, I forgot where it's native to, but it grows in very tropical type of environments and climates. So you find tropical Asia, they grow lots of sugar cane out in tropical Asia. Latin America, South America and stuff, they grow huge amounts of sugar cane out there. The United States, because it's not very tolerant of cold, you find it in the southern part of the southeast, Florida, Louisiana, and things like that. They have commercial sugar cane growing. But you're not going to find it up in, say, north central Alabama. It just won't tolerate the cold and what have you. So there's a lot of, and that's our major source of sugar is sugar cane. Now there's also a type of beet where the underground stem, the tuber, puts out and stores a large amount of sucrose. That's what they call the sugar beet, and that's grown up in the Great Plains states and stuff. And there's a lot of federal government support for these sugar beet growers, even though it costs far more than growing sugar cane and stuff, but hey, they got political clout, so you know they get what they want. Okay, so anyway, that's another but much lesser source and stuff, and that's mostly in the United States. It's not growing too much outside. Now, of course, sugar cane, aside from a sweetening agent, sugar cane, it stores it in the stems. And if you go to a place that has a high Latino population, a lot of times the stores will actually sell these sugar cane stalks. In fact, I think the one at Walmart in Oxford has them occasionally. And people take them and they just like suck on them like candy.
Sure.
You've done that. Okay, okay.
It's good.
But you were from New York. You can get anything in New York, right? But I think in Oxford they sell it there too. So it's kind of a tasty treat. Now, of course, the other thing is sugar cane is a great way of making ethanol. Nowadays, you know, 10% of what goes in your gas tank is ethanol in most cases nowadays and stuff. And ethanol is generally derived from corn. Now, the problem is when you make ethanol from corn, the energy input in fertilizer, growing the corn, shipping it out, processing it, all that kind of stuff is actually more than the energy output you get from burning the ethanol. The one advantage. And likewise, the carbon footprint of ethanol from corn is negative. In other words, it actually does more harm than good. Now, the one advantage of ethanol into corn, aside from making some farmers fairly well off, the one advantage of it, of course, is domestic. You don't have to buy it from a bunch of crazy people that hate us out in the other parts of the world. Yeah, they blow us up all the time, every time they get a chance. So anyway, you know, you get it domestically. But far better than that, and this is common in places like in parts of Latin America, like Brazil is trying to push this real hard. Brazil can grow sugarcane real easily, huge amounts. Real easy to take care of and things like that. So what they do in Brazil is they try to run as much of their economy on ethanol rather than on oil. Now they have fairly large offshore oil deposits that are just starting to be exploited now. But the idea is run your domestic economy on ethanol as much as possible. A lot of their cars run on E85, 85% ethanol, 15% gasoline. A lot of their cars and trucks and stuff do that. And because you can get a lot more ethanol from a sugar cane stalk than from a corn stalk, it's actually the kernels where you're getting the sugars for the ethanol. So it works pretty well. So the idea is run as much of your economy on ethanol and then sell all the oil that you suck out of the ground to us at a huge profit. So that's what they're trying to do, become almost energy independent themselves, and then sell the oil deposits that they're working on exploiting now and sell it to the U.S. and China and what have you and make lots of money. Okay, so that's one kind of disaccharide. It's the one we're very familiar with because, well, it's only produced by a limited number of plants. Now, another one we run into is something called two glucose molecules covalently bond together to form what's called maltose or malt sugar. Now here again, that's produced by a limited number of organisms. Maltose is produced by this grass called the barley plant. Germinating young barley plants store a lot of maltose in their stems and things like that. Now maltose, if anybody's ever had malted milk or malt balls, you know they're sweet, but they have a weird aftertaste. That aftertaste is due to the maltose itself. If you took a spoonful of straight pure maltose, it would taste about the same. Okay, and of course the other thing you can do with maltose is let yeast break this stuff down and ferment it, because barley malt is a major ingredient in beer, and of course if you distill it down in whiskeys and things like that. Okay, so that's produced by another limited range of plants. It's stored in this case, in the stems. Now there's another common disaccharide, and it's not made by plants at all. In fact, it's made by a certain group of animals. And that is lactose, otherwise known as milk sugar. And the organisms that produce and secrete lactose and other substances in this fluid called milk, by definition, are mammals. That's the definition of mammals. is they have these glands called mammary glands. Obviously, there's a bunch of other names for them. I can't. But I can't mention those in class. But anyway, they have these glands, these mammary glands, and the purpose of these glands is to produce and secrete large amounts of lactose, the disaccharide, lipids, and especially a particular type of protein called casein into a solution and use that to feed their young. And that was a major breakthrough in the development of life. That was one of the reasons for the success of mammals when they first came into being. Well, after dinosaurs were extinct, mammals have been around for about 210 million years. The dinosaurs dominated most of the global ecosystems in those days, but after they got wiped out, then the mammals quickly took over and became, for the most part, the dominant large animals in most land ecosystems, and even some aquatic ones, some marine ones. So one of the reasons for success, of course, is being able to produce milk, because that way, if you're a bird, for instance, you know, mommy goes out to feed and bring some food back to the young. But what happens if she doesn't find anything today? The kids go hungry. And of course, if that happens for a while, that's very, very bad. And the kids starve and die. Okay, on the other hand, if you're a mammal, even if mommy hasn't eaten today, she can still produce milk and feed her young. So the young have a much more reliable food supply that way. Now, eventually, if the mother is actually getting to the point of starvation herself, she will finally stop producing milk. Sometimes people going on extreme dieting and anorexics and things like that will actually stop milk production. But anyway, for the most part, provides a much more stable food supply for the young, and that gives them a much better chance of surviving at least their early childhood and stuff. So that was one of the major reasons for mammal success, the ability to produce this sugar-rich and fat-rich fluid and protein-rich fluid to feed their young. Now the interesting thing, even though glucose and galactose are both fairly sweet, the combination of the two in the form of lactose is actually not sweet at all. If you had a spoonful of lactose, it would hardly taste, you wouldn't be able to taste much of anything. It's just kind of a gritty type of mouthfeel and stuff. So lactose actually isn't very sweet in the least. Now a lot of people, of course almost everybody, unless you have a particular mutation, almost everybody as infants have the enzyme called lactase that can break the covalent bonds into the two sugars. But a lot of times people lose the ability to produce that enzyme. They shut the production of that enzyme down in adulthood and stuff. But some people can continue to make that. And it's kind of interesting. This is an example of relatively recent changes in certain human populations. The ability to continue to produce the enzyme lactase well after weaning and into adulthood and stuff. And people that don't, if they have something that has lots of lactose in it, like milk, they can't break the lactose down. Gut bacteria can and will, but the end results are upset stomach, bloating, gas, a whole bunch of unpleasant feelings. And that's lactose intolerance. But when you look at different groups of people, you find that the incidence of that varies quite a bit. For instance, in European populations, Europeans have been raising cattle and sheep and other things and drinking their milk for thousands of years. So people of European ancestry tend to have relatively low rates of lactose intolerance. If you go to people of Asian ancestry, most of them who did not herd cattle and drink milk and stuff, rates of lactose intolerance are quite high. You go to African populations and it varies because some African populations have been herding cattle for thousands and thousands of years and they have low rates of lactose intolerance, while other African populations never did that kind of stuff and they have very high rates of lactose intolerance. In other words, why produce an enzyme that you're never going to need again after mommy pushes you away from her breast? Well, that's what happened. So, on the other hand, if you're drinking some other animal's milk all the way into adulthood, there's going to be an advantage to continue to produce that enzyme so you can exploit that new food source. And those changes are believed to have occurred in the past maybe 10,000 years or perhaps less. They're relatively recent changes, biological changes in certain human populations. The ability to continue to produce the lactase enzyme all the way through adulthood. Okay, so those are a couple common disaccharides. And of course, there are many others, but these are some of the ones we're perhaps the most familiar with. All right, now we'll move on to polysaccharides.